The next video lecture, we're going to talk more about enzymes. All right, so enzymes are the workhorses of metabolism. You talk about enzymes in any biology class, and there's kind of an old rule of thumb. If you don't know the answer to a biology question, you can probably blame it on an enzyme in some way and at least get partial credit. And that just emphasizes how important enzymes are to the function of living things. All right, so we've already been talking about metabolism and how metabolism is the sum of all these chemical reactions that take place in cells, and metabolism can be divided up into catabolism and anabolism. So you have breakdown reactions and you have building chemical reactions. All right, so where do enzymes come into this story? So you've got all these coordinated chemical reactions that have to take place in living things. Could those chemical reactions happen randomly? Well, yes, they could happen randomly. So you could randomly have a couple of amino acids in a cell bump into each other and make a dipeptide. You could have a couple of monosaccharides floating around in a cell and they randomly encounter each other and they form a disaccharide. So those things could happen randomly. A protein could randomly break apart into individual amino acids. But those processes would be extremely inefficient, and there's no way that a cell, which has to perform all of these complicated, complex, coordinated chemical reactions to maintain itself for survival, would be able to survive if it was depending on a bunch of random chemical reactions. So enzymes are machines, chemical machines, and the vast majority of them are actually proteins themselves. So you have to kind of think about that for a minute. There are enzymes that build proteins, there are enzymes that break down proteins, but enzymes themselves are organic molecules and almost always proteins. And their role is to catalyze the chemical reactions in cells. And I want to point out this is not the same as catabolize. This is catalyze. It's a different word. They just sound very similar. And what catalyze means is increases the efficiency, forces the chemical reactions to happen, speeds them up, makes them much more likely to actually happen. So cells have many different kinds of enzymes that force all of these important catabolic and anabolic reactions to take place. Okay, and so generally enzyme names have the suffix ase on the end. And sometimes you can tell from the name what the enzyme probably does. So, for example, sucrase is an enzyme that breaks apart sucrose, or table sugar. Lipases are enzymes that break apart lipids. Now, I don't want you to think that enzymes, some students think that enzymes only break things down, and I think maybe that impression comes from Biology 202, when you take AMP2 and you learn about all the digestive enzymes that break apart proteins and carbohydrates and lipids in our diets. Yes, there are enzymes that do catabolic reactions like that, but you also need enzymes for your anabolic reactions, so do keep that in mind. For example, DNA polymerase. That's an enzyme that we'll learn more about when we get to Chapter 8 and we study genetics. DNA polymerase is an enzyme that builds DNA polymers. It builds DNA molecules. So it takes DNA nucleotides and bonds them together. But anytime you see ASE on the name of something, pretty good in biology, pretty good chance that that's referring to an enzyme. Another property of enzymes that's important, they can be reused over and over. So like sucrose takes sucrase, is an enzyme that takes sucrose. Sucrose is a glucose molecule and a fructose molecule bonded together, and it splits them apart. So sucrase can split apart a sucrose molecule and then go off and find another sucrose molecule to split apart, and then find another one, find another one, find another one. So they are reusable. If there's a slightly awkward transition in the video here, I accidentally skipped a slide when I was recording this video, and I'm having to go back in and splice in this one, so it might seem a little dicey around this particular slide. But let's talk about the components of a typical enzyme. This is another one of those things that you tend to talk about in lots of biology classes. So we've said that enzymes are proteins in general. Most enzymes are protein molecules. And that protein portion of an enzyme is shown here as the big purple blob, and sometimes that's called the apo. enzyme. However, enzymes often need other components in order to be able to function properly. And sometimes those are cofactors. Cofactors are usually metal ions, things like iron or zinc or copper. And you know, when you take a vitamin and mineral supplement, or you look on a box of cereal and they list all of those vitamin minerals, the reason we need all these, remember we talked about micronutrients, like those metal ions in our diets, is because they are used by one or more enzymes as cofactors. So those cofactors, and again those are usually ions of some type, need to be there in order for the enzyme to carry out its chemical reaction properly, and or it may influence the overall 3D shape of the enzyme. Coenzymes, on the other hand, these are generally larger organic molecules, not as big as a protein, but they are organic molecules that also participate in the chemical reaction. And they're often vitamin derivatives. So the reason that we need vitamins in our diet, as well as other organisms needing vitamins, is that many of those vitamins are the organic molecules that get converted into coenzymes that are used by one or more protein enzymes inside cells. So that's some terminology to keep in mind. Now, not all enzymes have cofactors or coenzymes. Some don't have any, so it would just be the apoenzyme or protein portion. Some enzymes may have a cofactor, some enzymes may have a coenzyme, some enzymes may have both. Lots of different enzymes, and these things vary across a whole spectrum. If an enzyme requires a metal ion cofactor, or it requires a coenzyme in order to function properly, then it's got to have those things, otherwise it won't work. So like we said, that's why you got to have these vitamins and minerals in your diet, is because you have one or more enzymes that requires them, and if you can't make these things on your own in your body, then, and if your enzymes aren't working, you're not going to be a happy, healthy organism. Okay, so let's talk a little bit more about enzymes and what they do. So here in purple, we've got a generic enzyme. Keep in mind that big purple blob there is almost always going to be a specific protein whose job is to be an enzyme. And what this diagram from your textbook is trying to show you is that particular enzymes are very specific about what they act on. Okay, so enzymes will have a pocket that has a specific three-dimensional shape that binds something that it's going to act on. And whatever it binds to is its substrate, or it could be substrates. Some enzymes are going to bind more than one thing to force a chemical reaction to occur. Okay, but that's something to keep in mind, that whatever an enzyme attaches to and is going to do something to, that is called the substrate or substrates of the enzyme. And what they're showing you here is we've got a couple of generic substrates over here. So these are other types of molecules. I know that one looks like the nipple on a baby bottle, or maybe a plunger. I don't know. I'm not sure what that's supposed to be. But these are supposed to be two different molecules. And notice that this one fits into the catalytic or the active site. That's the little 3D shaped pocket on the enzyme. And this other one does not. So they're very specific about what they bind, so they're going to only act on what they're supposed to act on. When those two come together, you have an enzyme-substrate complex. And here the enzyme is forcing whatever chemical reaction to take place to occur. And then finally, the enzyme releases the product. So in this case, we had a substrate which was a larger molecule, and it got broken apart into two products, which are smaller molecules. So would the chemical reaction performed by that enzyme, is that a catabolic or an anabolic reaction? Hopefully you're thinking catabolic, because you're taking a larger molecule, like this might be sucrose, and sucrose would be the substrate of that enzyme, and it got broken apart into two products, which in this case would be glucose and fructose, are the smaller monosaccharide products that come from breaking apart sucrose. So whatever an enzyme attaches to and reacts and will act upon, that's the substrate or substrates. And then whatever the enzyme has made are the products. The inputs of the reaction, substrates, outputs, products. Okay, and again, so what's the role of enzymes? So could that molecule just spontaneously break apart into those two products? Yes. But an enzyme forces that reaction to take place. So in a sense, it's almost like it's breaking apart the molecule and forcing it to happen. All right, and here's another look. This is a little bit more of a detailed look at an example enzyme. Here we have a molecule called fructose 1,6-bisphosphate, and there's its chemical structure. And so that's an organic molecule. And this is actually a molecule that is part of the glycolysis pathway that we'll talk about later in this chapter. And here's an enzyme whose job it is to split that molecule into two smaller ones. So you see the enzyme attaches to this. This is the substrate of the enzyme. That forms the enzyme substrate complex where those two are together. And then the enzyme forces that molecule to break apart, and it breaks it apart into these two smaller molecules, glyceride 3-phosphate and dihydroxyacetone phosphate. So those are going to be the products. And then notice the enzyme gets released, so it can be reused again. It can find another fructose 1,6-bisphosphate to attach to and break apart. And in all likelihood, this is the only thing this enzyme does. Enzymes are very specific about the chemical reactions they perform. So here's your input, that's the substrate. Here are the outputs, those are the products. So you want to be able to look at a chemical reaction that an enzyme performs and be able to state what is the substrate or the substrates and what are the product or products of that chemical reaction. So for example, if you've got a DNA polymerase enzyme, we talked about that, and it takes nucleotides and bonds them together, what's the substrate or substrates? The substrates will be the nucleotides that are going to be bonded together. So that DNA polymerase enzyme is going to bring those two things together, force them to bond, and then that's the product. The bonded nucleotides are the product of that particular enzyme. All right, enzyme activity. Enzymes are affected by various factors, so this is something else that you hear about in every biology class. One of those is temperature. So on these graphs, they're showing you enzyme activity on the y-axis, and then something else that impacts the enzyme on the x-axis. But enzyme activity, what that's referring to, an enzyme's activity is measured by how rapidly, how efficient it is at making its product, finding substrates, and making products. And so this is showing you that enzymes have optimal temperatures. Remember how we talked about how organisms have optimal growth temperatures? Well, you will find that those optimal growth temperatures parallel the optimal temperatures of that organism's enzymes, because in order to be able to grow and reproduce, you have to do your chemical reactions, you have to do your metabolism, so therefore your enzymes have to be working optimally. So enzymes will have an optimal temperature at which they work best, and just like with growth of an organism, it drops off very quickly as the temperatures go higher, and it drops off more slowly as the temperatures cool off. If temperatures go higher, this is supposed to be a diagram of a fully folded functional protein, and enzymes are usually proteins. And over here we have a denatured one that has unraveled and lost its three-dimensional shape. So if you remember that enzymes have a specific product pocket, a catalytic site or an active site that substrates have to fit into, that 3D shape is very important. So if an enzyme denatures and loses its 3D shape, it can no longer bind to its substrates and it can no longer make its products. So temperature is absolutely critical for the activity of an enzyme, because if temperatures go too high, proteins denature. pH influences enzymes as well. Same thing. If you go outside the optimal pH level for a protein, proteins will denature. Another thing is substrate concentration. This is kind of common sense. Enzymes have to be able to find their substrates in order to perform the chemical reaction and make the products. So what this is showing you is that enzyme activity, and again, enzyme activity is really referring to how efficient the products are made. So substrate concentration will impact that. So if your substrate is sucrose, because you're going to break apart sucrose, it just makes sense that as sucrose levels go up in a cell, you're going to make more of the products of that enzyme sucrase, because there's more sucrose around for those enzymes to bond to and make those products. Eventually, though, you hit a saturation point, and what that means is eventually you've only got so many of these enzymes available in a cell, and eventually you're going to saturate all of them. They're all going to be involved in making products, and the only way you would be able to make more or have a higher level of activity would be to have more of the enzymes around. So temperature and pH changes can make enzymes labile, which means they denature, they are no longer, they unravel, they're not able to do their jobs. The concentration of the substrate and the concentration of the enzyme. If you have more of a particular enzyme around, you're going to be able to make more of the products. If you have more of the substrates around, you're going to be able to make more of the products up to a certain point. Okay, another concept with enzymes is that the activities of the enzyme, how well they are making their products, that needs to be controlled. And that's generally controlled in cells through inhibition of some type. So cells are very careful about making only as much product as they need. So cells try not to be wasteful, because being wasteful uses up energy, and energy is very valuable to living things. So in general, cells are very careful about only making the amount of a product that's needed. All right, so enzymes, their activities have to be controlled. You know, at times you want the enzymes making products, at times you don't. And there are a couple of different ways that can be done. Now, one is that you don't make as much of the enzyme anymore. If you don't need the products, cells make their own enzymes. They're proteins, so they manufacture their own enzymes. So cells can control the amount of those enzymes that are around. Existing enzymes, though, have to be inhibited to reduce their activity. And that can occur through competitive inhibition. And what that means is that there's some kind of inhibitor molecule that binds into the active site of the enzyme. And that's what they're showing you over here. So we've got a generic enzyme, and this is the normal substrate of the enzyme that would fit inside that catalytic site. And over here we have an inhibitor molecule of some type that has a very similar shape to the normal substrate and fits down in that pocket. And so if you have inhibitors bonded inside this enzyme, the normal substrate can no longer attach. And because of that, over here on this side, you're not making any products. Here's your products over here. You're not making any because the enzymes are blocked. Another way that enzymes can be inhibited is through non-competitive inhibition. And that means that whatever the inhibitor molecule is, it doesn't attach in the active site of the enzyme. Instead, what it does is it attaches over to a separate little area on the enzyme. And when it attaches, so our inhibitor here looks like a dumbbell, looks like a bell, not a dumbbell. That molecule is attaching to the enzyme, and when it bonds, it warps the enzyme. It causes the enzyme to change shape. And your little catalytic site over here, that little pocket changes shape, and it can no longer bind the substrate. And if you can't bind the substrate, you're not going to make any more products. And interestingly, what they're showing you over here is that in many cases, the product of an enzyme inhibits its own enzyme. And that's a type of negative feedback like you guys learn about when you study homeostasis in anatomy and physiology. And you especially run into that when you cover the endocrine system in biology 202. The products go back and inhibit the very enzyme that manufactures it. And that makes sure that you don't make too much of a product. You don't just keep cranking it out over and over and over again when you really don't need any more of it. A lot of times it's not quite that simple. This is another diagram which is showing you a product down here at the bottom of a reaction pathway. So very often catabolism and anabolism occurs not through just one chemical reaction, but through a series of chemical reactions, step-by-step reactions. And that's what they're trying to show you here. We're starting with a substrate molecule of some type, and we're ending with a product down here. But it takes 1, 2, 3 steps, 4 steps to actually make that product. And each of those steps has its own enzymes. I'm sorry, there are 3 steps because we got 3 enzymes. And so that's what they're trying to show you here. The original substrate attaches to enzyme number 1, and that makes a product that's called intermediate A. Intermediate A attaches to enzyme 2, and it gets changed, and that makes product or intermediate B. And then this one attaches to enzyme number 3, and finally we get our end product, or the final product that the cell was trying to make. So a multi-step process, but notice this end product can go back up here and bind to enzyme number 1, this one. So it binds into that other little pocket that causes the enzyme to warp or change shape, and now enzyme number 1 can no longer attach to the original substrate in that pathway. So it's going to shut down that whole biosynthesis pathway. Or it could be it doesn't necessarily have to be a synthesis pathway. It could be a breakdown pathway that's taking place as well. All right, so what's the advantage here? Again, if you think about it, if a cell has to go through a multi-step process to accomplish something and use multiple enzymes, yeah, you could shut down the 3rd enzyme, but that means enzyme 1 and enzyme 2 will still be performing their chemical reactions. And it's more efficient if you don't need this product. Well, let's go back up here and shut down enzyme number 1 so we don't waste resources going through all of those other steps as well. All right, from a clinical standpoint, a lot of the drugs that have been developed and that we take for various things, not just infections, but other things as well, work through inhibiting one or more enzymes. For example, when you take penicillin or its many relatives as an antibiotic type drug because you have a bacterial infection, penicillin inhibits an enzyme that manufactures the peptidoglycan cell walls of bacteria. And when bacteria can't make their cell walls, remember they're prone to bursting due to osmosis. They uptake too much water. Another drug offhand that I'm thinking of, acetaminophen, and many of the other drugs that you take for pain relief inhibit enzymes that are involved in the production of signaling molecules called prostaglandins that are used in many different ways in the body. So if you inhibit the enzymes that make those prostaglandins, the prostaglandins are no longer able to signal things like pain or fever production. That's why you can take Tylenol and it will reduce a headache and also cut back on a fever as well. All right, so that is just some background information on enzymes, because as we talk more about catabolism and anabolism as we proceed through Chapter 7, you need to keep in mind that enzymes are always involved. And another thing to think about, too, at the microorganism level, different microorganisms have different repertoires of enzymes. They have different sets of enzymes that they use. And if an organism has a particular enzyme, it's going to be able to carry out the chemical reaction that takes place. If it's lacking a particular enzyme, it's not going to be able to perform that chemical reaction. And that winds up being important as you guys move into your Learn Smart Labs activity on identification of unknown bacterial organisms, because many of the tests that are used to identify bacteria are based on enzymatic activities. Does this organism have the enzyme that allows it to break down this nutrient or make this product? Those types of things can almost always be traced back to the enzymes that organism has. So keep that in mind as you go through the rest of the material in this section of the course. And on the next video lecture, we're going to get more into how organisms harness energy in order to be able to survive.